All right, time to go teach. Hello and welcome to this next episode of Tutors, continuing our lessons on the cell. And today we are continuing from the last lesson, which was talking about the cell cycle, to being in S phase, looking specifically at what happens during that phase. And I realize in that lesson, I didn't actually define what the S in S phase stood for, and it stands for synthesis. So we are going to be looking at DNA synthesis, which takes place during the synthesis phase, or the S phase. So today, targets. First off, we're going to compare and contrast the difference between how eukaryotes and prokaryotes replicate their DNA, because they do it slightly differently. And then we're going to identify the different proteins that are going to be involved, what functions they perform, and how they go about doing that process. And lastly, we're going to describe how the actual process of DNA replication occurs. We're going to go through it step by step on how each of the different enzymes is used to show how the DNA gets replicated. Let's get this thing started. So first, it all begins at this spot called the origin of replication. And for prokaryotes, they'll only have one origin of replication. They only have one chromosome, and so that one chromosome will have one origin of replication. On the other hand, eukaryotes, each chromosome can have hundreds of origins of replication. So it can be replicating in many different spots. And many of the eukaryotic cells have multiple chromosomes. So each chromosome can be replicating in many different parts of itself at one time. And again, for a eukaryotic cell, they have this specific phase, the synthesis phase, which is where the DNA is replicated. So an origin of replication would be where the DNA is actually pulled apart and our new strands of DNA are going to be built. So each one of those would be a replication point. And in this case, this little strand would have had 3 different origins of replication. Some of the proteins that are involved. First, helicase. Helicase is going to be involved in breaking the hydrogen bonds between the strands of DNA. So remember that each strand of DNA, its nucleobases, are held together by those hydrogen bonds. The adenine to the thymine with their 2 hydrogen bonds, and the cytosine and the guanine with their 3 hydrogen bonds. Helicase will separate out those nucleotide pairs. Topoisomerase, it will help stabilize this DNA structure as replication is taking place, as will the single-stranded binding proteins. Some other enzymes that are going to be involved. DNA polymerase, that is going to be involved in the process of elongation. Elongation is the term that we use for the synthesis or the building of our new DNA strand. DNA primase is going to be involved in actually building a little primer that is required to actually start the process of elongation. The DNA polymerase 3 cannot build the DNA strand without a little primer being attached first. It needs a little bit of RNA so that we can build DNA. And we go to DNA polymerase 1, which is going to remove those RNA nucleotides and replace them with the DNA nucleotides. And ligase, DNA ligase, which is going to attach or join together, using dehydration synthesis, these segments of DNA called Okazaki fragments. We'll talk about how those fragments appear, and then, obviously, ligase is going to join those fragments together. So, let's look at the process as it takes place. If this is our strand of DNA, we have our 5' ends, we have our 3' ends. They're anti-parallel to each other, so we can look at this strand of DNA and see that we have these different directions. Helicase will come in and separate out the strands of DNA from each other, breaking those hydrogen bonds between the different nucleotide bases. As it moves through, primase is going to be able to add RNA nucleotides that are going to be complementary to the template strand. And you'll notice that it moves in the 3' to the 5' direction. It cannot go in the opposite direction. Primase can only move along the template strand in the 3' to 5' direction. And that means that this bottom strand, primase was able to move along and build an RNA primer. But our top strand right here, the end that is open, is a 5' end. And the RNA primase cannot move in the same direction as a helicase is moving. So that's going to have a big impact upon how we build our DNA molecule. As helicase continues to move across, now DNA polymerase, because there's a primer added, can start to move across, following along as helicase moves. This bottom strand right here, where the DNA polymerase is moving in the same direction as a helicase, is called the leading strand, because the helicase is moving along that strand in the 3' to 5' direction. The DNA polymerase can then follow right behind the helicase and perform the function of elongation. On the other hand, this top strand here, the helicase was moving along it in the 5' to the 3' direction, which is the exact opposite direction that both primase and DNA polymerase III are able to move. So, after helicase has moved along and separated out these strands sufficiently, a primase is able to bind with this top strand of DNA, and now it will move in the 3' to the 5' end. So it will move along 3' to 5', building the primer. DNA polymerase can move right along behind helicase here. Now that a primer has been added on this top strand, you can see that DNA polymerase is able to build on that. We have to add a new primase in on this top strand, and it will put a primer in, and now DNA polymerase will come in to add more DNA nucleotides. And this process will continue. Now we're going to add some nucleotides right here for DNA polymerase, adding those nucleotides in. And now we have our strands of DNA. Now instead of having only one copy of our DNA, we have 2 copies of our DNA. 1, 2 copies. The primer must be removed, so DNA polymerase is going to come in and it will remove the RNA nucleotide and add in DNA nucleotides that are appropriate. Something to notice here is that we have gaps on this top strand. Our thymine and our guanine nucleotides right here are not attached. Our guanine and our cytosine nucleotides are not attached right here. These are different fragments. We have a leading strand on the bottom. Our top strand is referred to as a lagging strand. I did not mention that before. And the lagging strand is built in the opposite direction that the helicase is moving. Okay? Still in the 3' to 5' direction, but it is in the opposite direction of how helicase is. Because of the anti-parallel nature, you're always going to have one leading strand and one lagging strand. Because the lagging strand had to be built in segments, we end up with these fragments that are called Okazaki fragments. And that is where DNA ligase is going to come in and join these fragments together. So our ligase enzyme will come in and build that bond between those nucleotides. So now they are all joined as one. That is the process of DNA replication. It always starts at the origin. Now, I showed it in the previous little example where it started from one end, but a lot of times it's actually going to start in the center of the DNA. And so if we have our origin on both sides where the DNA gets separated out, we will see that you will have elongation occur with the leading strand going in the 3' to the 5' direction and the 3' to 5' direction. And the lagging strand is also going to be on both sides because it has to be built in that 3' to 5' direction, and it will be built in segments, those Okazaki fragments. So that's how we have our lagging strands. The results of this is our two strands are copies of DNA, but neither copy of DNA is all original or all new. Each copy of DNA contains a template strand and the newly synthesized strand of DNA. So, because of that, we call it semi-conservative replication. Neither one is wholly conserved, neither one is brand new either. They are both old and new, each copy containing one template and one newly synthesized strand. So in summary of all this, all cells, they have to replicate their DNA. And that process is always going to follow some very specific steps. The eukaryotes are only going to ever replicate their DNA in the S phase. In the synthesis phase of the cell cycle is when eukaryotes replicate their DNA. The DNA polymerase, it can only move in that 3' to 5' direction. It cannot move in any other way along the DNA strand. You're always going to have one strand that's a leading strand, and the anti-parallel strand to it must be the lagging strand. The lagging strand will have the Okazaki fragments, which will have to be joined together using DNA ligase, and the leading strand, the DNA polymerase, will have followed along right behind helicase the entire stretch as helicase moved along that strand in the 3' to 5' direction. And that is DNA replication, or DNA synthesis, which takes place during the S phase. Until next time, be awesome, stay awesome.